Why are you asking about that?
All right.
Just in case I want to like Photoshop stuff onto talking later.
Okay, we're recording it now.
We are
Like
recording.
cool glasses or something.
All right, Vlad,
Let's
go
be
ahead.
serious. Okay, so I guess I can start.
You can introduce yourself first.
Yes, I will introduce myself with sharing my screen, and just a second. Presentation. There are a lot of slides. Okay, can you see it? Awesome. So hi everybody. My name is Vladimir Bajić. I'm originally from Serbia. I did my bachelor, in Belgrade and one exchange year in US. And after that, I started my MEME program. So for the first semester, I went to, let me just minimize this in case it's over your screen. I went to Uppsala, and there I did this very small project with Matthias Jakobsson. I was basically interested in languages and how languages could be used in human population genetics. For the second semester, I got interested into tits, great tits. So that was a research project in at LMU. And it was basically a lot of cycling through the city. It was very interesting. I wanted to do a project to just go around and explore the city or to be more in nature. So we were looking at the behavior of the tits. For my first master thesis, I went to Harvard in Hextra Lab, and there I was working with Andres Bendensky, where we were trying to use genetic maps to basically improve the quality of the assembly of the Permiscus maniculatus genome. It's a small mammal like a mouse in Europe. And for the fourth and the best semester ever, I was in Montpellier. So I think that was also the semester when I learned the most and one of the most interesting projects I had. So that was the topic about evolution of the pygmy mouse feminizing X chromosome using genomics. They had two individuals sequenced, one that has a very specific X chromosome that is leading to feminization of XY individuals. So during MEME, I realized that I know that I don't want to do my PhD in US, and I don't want to do my PhD in Germany because it was so terrible in Munich and I didn't like it. People didn't want to speak English to you. They were rude and stuff like that. But then I started searching for positions, and guess what? I ended up in Germany. So I found a PhD position in Leipzig, which is a small, relatively small city south of Berlin. And it is known because of Svante Pääbo, who was the first to sequence a Neanderthal genome. And this actually gave a lot of glory to the institute itself. But I haven't been working with Svante himself, but rather with Mark Stonking, this person here, who is known for and got established but mostly known for this out of Africa theory, together with Rebecca Can and his supervisor, Alan Wilson. They published a paper using mitochondrial sequences, and they showed basically that the highest diversity of mitochondria is within Africa, and that this diversity declines the further you are from Africa. So he was my supervisor, and of course, the work would involve a lot of mitochondria. And now I'm doing my postdoc in Katja Novik's group. It's in Berlin, so not that far away from Leipzig. And here, my reality is a bit different. Most of my time I've spent so far teaching myself to teach. And the little bit of leftover time that I have, I'm actually trying to do actual science. And it's going very slow, unfortunately. So today I will be talking about my PhD project, which was titled as New Approaches to Investigating Sex-Biased Admixture in Southern African Population. So as I said, my supervisor was Mark Stonking, and this work was done at the Department of Evolutionary Genetics at Max Planck Institute in Leipzig. At the very beginning, I would like to start with giving you a short background story. So what are the commonly used genetic markers in human population genetics? And then I would introduce 2 chapters for my thesis. The first one is focusing on the genetic structure and sex-biased gene flow in the history of Southern African populations. And the second chapter is sort of a natural prolongation of the first project, where I sort of got some ideas, and I was thinking to start integrating different genetic markers into single story, trying to infer ancestry packages, or in other words, mitochondria and Y chromosomes that are associated with autosomes. So for the very beginning, let's start talking about very commonly used genetic markers. So one of them is the Y chromosome that is inherited patrilinearly. That means that father transmits the Y chromosome to his sons and only sons. On the other hand, and of course, if we are going to human populations or any other populations that have Y chromosome, right, we can investigate it and get information about paternal history of the population. And it does not necessarily have to be true for the entire population itself, right? Then we can look at the mitochondrial DNA, which is inherited from mothers to her offsprings, regardless of the sex, but it's matrilinearly inherited. And it's much smaller in the size, it's different cellular localization, different mutation rates, and so on. And finally, what we can do is to use autosomes, but that I'm going to mention in a few slides. So why mitochondria and Y chromosomes are cool? Well, because they represent a single haplotypes. They are linked, there is no recombination on them, well, for the biggest part of the Y chromosome, and thus we can see them as haplotypes. And here, just as an illustration, I show you 5 different haplotypes that differ in SNPs that I colored here. And by carefully inspecting differences between haplotypes, we can go back and trace when each of the mutations occurred and reconstruct the tree of the haplotypes of those sequences. And by looking which of those SNPs are shared between different haplotypes, we can define arbitrarily the haplogroups, which are going to be basically groups of haplotypes that share a common SNP. And this is true for mitochondria and for the Y chromosome. And this is something that people in human population use a lot, and we're referring to haplogroups by their names a lot. And here is example of mitochondrial phylogenetic tree. This is from the review from 2007, which I really like a lot. And what we can see here is that, so here we have a time scale, and below we have colors that are designating where each of those lineages, those are names of haplogroups, are occurring. So we can see that lineages that are deepest splitting are occurring within Africa, meaning that the highest diversity is within Africa, and only a subset of this diversity, represented with the branch of L3, went outside of the Africa, which basically recapitulates the event of out of Africa. And the further time went through, we get separations between finer scales of geography. And so if we would go more recent in times, for example, at k=6 here, we would see that lineages that are occurring within Americas in this dark purple or whatever, magenta, I don't know really the name of the color, we can see that it's splitting from the lineage that is closely related to those found in East Asia, and which is true for the others also that are found in Americas. So basically, this shows that mitochondrial sequences are indeed useful to track human migrations. And the very, very similar story applies for the Y chromosome. Again, we have the deepest lineages found in sub-Saharan Africa. Only a subset of this diversity exited, went out of Africa. And again, for example, in Americas, we see lineages that are sort of related to those found in East Asia. And timing of those differences sort of correlates with the timing when the humans arrived in different geographic areas. And autosomes are basically a crown of the research because they harbor the most information. Majority of the SNPs you can find are going to be on the autosomes. They are biparentally inherited, unlike mitochondria and Y chromosome. What does that mean? It means we are inheriting it from both parents. And this is going under recombination. So each of our parents has 2 copies, and those copies, when inherited, they are inherited in such a way that we are not inheriting exactly the same chromosome. There is recombination, meaning that after a few generations, we are not going to have information only about patrilineal or matrilineal side, but actually from much bigger set of ancestors present in our ancestry tree. And having such a diverse and complex data, and indeed very, very big data, there are several tools that human population geneticists developed to summarize this diversity and to investigate populations. So here I'm showing you admixture. I'm sure that most of you, if not all of you, are very familiar with such kind of analysis. And here we have a map of individuals from very famous project, HGDP, Human Genome Diversity Project, started by Cavalli Sforza, who had the idea to capture diversity of humans around the world. And if you would run a mixture on individuals from those populations, we would get a plot that would look something like this here at the bottom, where we would have populations and each vertical line would represent a single individual. And then we would be able to divide genetic composition of a single individual into desired number of ancestral clusters. In this case, we defined it at number 8. That's why we're going to have 8 different colors. And if we would look how those colors are distributed across the globe, we would see interesting pattern, expectedly. Those that are more similar in color are sort of closer geographically. So we have, for example, those that are purple, dark purple are in Americas, those that are pink are in Eurasia, and so on. And I have to say that this admixture is very, very commonly used. So PCA and admixture, they're basically like on the fast track in human pop gen journals. So everyone is using it. And often it would be beneficial to know how different mitochondrial haplogroups and Y chromosome haplogroups are relating to those. Someone wants to join, how they're relating to those admixture components that we infer using admixture. So in the first chapter of my thesis, or the only paper I actually have from my PhD, I investigated genetic structure and sex bias gene flow in the history of Southern African populations. So my supervisor, Mark Stoneking, and his wife, Bridget Packendorf, went to Southern Africa and they collected data from a lot of populations. But why Southern Africa is so interesting could be summarized in this plot here. If this is Africa, weirdly positioned. And here we have a phylogenetic tree of human populations. And if you see here in red and purple are Southern African populations that are included in my study. Those are Southern Khoisan individuals and Northern Khoisan individuals. And we can see that they capture actually the first split in human populations. So the deepest split is between them and everyone else, including other African populations and non-Africans. And you can see the diversity outside of Africa is minusculous compared to what we see within Africa. So investigating such a diverse population and deeply divergent population is very interesting from evolutionary perspective. So when we're talking about Southern African hunter-gatherers, we use term Khoisan that actually means basically different people that speak languages that can be characterized with click sounds. And those click consonants actually are here. And this is the part when I embarrass myself and try to pronounce them. So we have 5 of them. So we have dental click, for example, in Gui population, or lateral click is Ghana, or Juan, or I don't know how to do this. And I mean, it's not that I know to do any of them, but I have to fake it a bit. It's very interesting. If you don't know how that really sounds, I recommend you to go to YouTube and search for Khoisan speaking. It's really interesting. So they can be divided in 3 linguistic families, colored in different colors here, in blue, green, and red. And there has been previously research on them, but what hasn't been done previously is investigating paternal history of those populations. What we know from literature is that there are at least 2 admixture events in Khoisans. First is related to populations that developed around 10,000 years ago in the Middle East. Those populations developed agriculture. They expanded agriculturally. They reached Eastern Africa around 3,000 years ago and admixed with them. And as such, they moved further south, reaching Southern Africa around 2,000 years ago. And we know that modern Southern African Khoisan populations harbor between 9 and 30% of their genomes related to this admixture event. And this we know not only from autosomes, but also from other genetic markers, such as lactase persistent variant, which is a mutation in the lactase gene that is associated with the ability of the adults to digest milk, unlike me. So the prevalence of this allele is highest in, for example, Northern Europe, and people from Southern Europe tend to have to be non-lactase persistent, and I'm Balkan. So another example comes from the Y chromosome, E1B1B, it's called, and the marker mutation often is used, which is M293. So there are several lines of evidence that suggest that there was migration from the East. And more obvious example of admixture comes from a population that started expanding from West Africa. So in Nigeria-Cameroon border, around 4,000 years ago, people also developed agriculture, later on also smelting technologies and iron use. They started expanding all over the Africa. And this is actually known as Bantu expansion, which resulted in incredible unification, genetic unification of most part of African continent due to the admixture and expanding populations. They also spread their languages, and that's why Bantu languages are the most widely distributed within Africa. They reached Southern Africa around 1,500 years ago, and of course admixed with Khoisans. And this is still ongoing process because Bantu pastoralists and agriculturalists are still expanding their territories within Southern Africa. So the aims of my study were basically to investigate paternal genetic history, which so far hasn't been described, but also to look at the sex-biased gene flow between incoming groups and local Khoisan people, which from the literature we know should occur. So for this, I collected the data from this region here, and those are the populations. Again, I keep the colors more or less constant through the talk, where in red, green, and blue, I'm going to have Khoisan populations, and in purple, I have Bantu populations. And we can see that nowadays distribution of Khoisan populations is more or less limited to the Kalahari Desert basin. And this is due to the fact that once agriculturalists are expanding, they're looking for area where they either can grow crops or just sustain the cattle. So hunter-gatherers were forced basically to move to the area which is less favorable for agriculture. So for this, we had a sequence captured actually around 900 kilobases of the Y chromosome non-recombining part for around 500 individuals. And this was previously published in the paper from my colleague in 2016, but they haven't described the paternal history in detail per population. I also collected mitochondrial sequences from my colleague for the same set of populations. And this resulted in overlapping 23 populations shown here, from which 17 are Khoisan and 6 are Bantu populations. Yes, in order to investigate the sex-biased gene flow from broader perspective, I also included publicly available data sets and frequency estimates for wider range of populations from Southern Africa. So the first thing was basically just to look at the distribution of haplogroup frequencies. And here we have a simplified Y chromosome tree. And here we have its distribution across populations. And the first thing one can do with such a data set is basically to run, for example, correspondence analysis and see which haplogroups are in higher frequencies in which of the populations. And here, just by eyeballing, we can see some kind of separation between those in purple that are Bantu speakers, so agriculturalists and pastoralists, versus those that are in other colors that are Khoisan speakers. And we can also notice that populations that are blue are widely distributed across the plot, meaning that it might be admixture. So similarly, but quite differently for mitochondria, we see that almost all of the Khoisan populations are highly grouped together due to the fact that they have very, very high frequencies of L0D and L0K. Those are haplogroups that are in literature already known to be associated with those hunter-gatherers. And we have a little bit more diversity seen in Bantu populations. So once I had frequencies of Y chromosome, I could ask myself, okay, if I would have to classify those chromosomes, haplogroups into Bantu or Khoisan, how it would be? Well, what I can do is basically look at the distribution of frequencies of those haplogroups within Khoisan populations, shown in light bars here, and in darker are their frequencies in Bantu populations. What we can notice is, for example, that those that are in green, they occur in higher frequency in Khoisan populations, meaning that they're probably associated with Khoisans, while those, for example, in purple are associated with Bantus, as they occur in higher frequencies there. Why it's not exclusive? Because of the gene flow, of course. And here I actually colored them according to the literature. So we know that through literature, those were described previously as Khoisan related. The one in gray was previously described both as Khoisan or Bantu or both. So we don't know actually. And throughout the analysis, I treated these as both. And in Bantu, we see those haplogroups with E. This one was previously associated with migration from East Africa. And then we have a set of haplogroups that are probably brought by European colonists in the region. So once I could classify haplogroups as Khoisan or Bantu, or Khoisan versus everything else, I could look at the sex-biased gene flow. So here I have something I call a diamond plot, where I'm showing basically few things. So at the very top of the graph here, we are expecting to see populations that are going to have exclusively non-Khoisan haplogroups for both mitochondria and Y chromosome. While on the bottom, we would expect to see populations that are going to have exclusively Khoisan haplogroups. And the closer they are to each of the ends, we would expect to see stronger sex bias, meaning most of the males are harboring non-Khoisan Y chromosome, and most of the females from population here are going to be carrying Khoisan mitochondria, and vice versa on this side, right? It's basically plot showing the differences in frequencies between Khoisan versus non-Khoisan for both mitochondria and Y chromosome. And the interesting thing for our data set is that almost all of the populations are falling in this part of here, meaning that all of the populations are showing the same bias, where females are predominantly carrying Khoisan mitochondria and Bantu Y chromosome. We also can see that populations that are blue tend to have stronger, maybe, sex bias, and also Khoisans in general compared to Bantu populations. So if I would look at a broader scale, so here I included populations for which I could find mitochondrial and Y chromosome frequencies, we see interesting pattern. First of all, there is increase of sex bias intensity. So here is basically measuring sex bias intensity. The darker it is, the stronger discrepancy is between mitochondria and Y chromosome. The more mitochondria from Khoisan and less of Y chromosome that is Khoisan population has. So meaning that they are strongest here and here. And if we would plot it based on the north to south gradient, we could see that further south you go, the stronger sex bias gets. And also we can see that sex bias is stronger in Khoisans and less strong in Bantu populations. So why is this? There are several hypotheses that could explain it, and anthropologists actually know already that there are several taboos in culture, both in Khoisan and Bantu, that prevent or encourage certain marriages. For example, if you're Bantu, you're of course encouraged to marry female or a male from the same population or someone who speaks Bantu languages. If you're Khoisan, of course. If you're Bantu male, you can marry Khoisan female, and often there are sort of kind of myths that Khoisan females are more fertile. They are maybe seen as sexier because of their curves and more fertile, and thus might be preferred by some Bantu males. While marriages between Khoisan males and Bantu females are forbidden by Bantu rules. So the gene flow in this direction would almost never occur because of social restrictions. Another thing that plays a role is basically, for example, for a Bantu male, is that the fact that if he wants to marry a Bantu female, he would usually have to have some kind of bride's pay, or I forgot the real term, but it's like a gift for the bride that you have to pay to her family. And this is usually a cattle for example, pastoralist populations. And it costs a lot, right? While if you would like to marry a Khoisan female, which is coming from hunter-gatherer society, they don't have this capitalistic way of looking at material stuff. They live very simple, and they don't tend to possess private things, but rather it's community-based. So he wouldn't have to give anything except his love for her. So that could be one of the reasons to prefer. And the fact that we see it actually geographically structured could also reflect the fact that sex bias changed through history. So populations on the north, we can imagine, arrived there, Bantu arrives there before they move south, could reflect the fact that they were depending on the knowledge of the local populations to survive in new environment, or that those norms and social taboos to marry Khoisans would evolve later on through the history. So this might reflect actually the timing of arrivals of Bantus. So the more recently they arrived to the south, the stronger taboos they had against Khoisans. So what I could conclude from the first chapter, am I too long speaking? No, is that there is very complex population history of Southern Africa, that Bantu expansion actually led to very strong sex-biased admixture, which is probably a result of interactions between Khoisan females mostly and Bantu males. And that this geographic pattern that we can see in sex bias probably reflects the changes in the social dynamics between Khoisans and Bantu groups over the time. So for the second project, I decided now to look a bit more systematically into those different kinds of markers that we're using in human population. So I had already data that included mitochondria and Y chromosome, but I didn't really look at the autosomes. And if you go through literature, they're often referring to autosomal ancestral components. And we would tend to believe autosomes more than the Y chromosome. So this is currently under review. I just got a few days ago a bunch of reviewers' comments that are asking for simulations. So if any one of you is interested in collaboration and is good with simulating, please just write me and let's have a talk to see if you could contribute. And I would try to explain in a few minutes now what is my idea. So what I'm trying to do is basically to unify what in human population genetics we often do very, very separately. So we go to the field, we collect the data, we extract, for example, mitochondria and investigate maternal history of population. Then we decide, okay, let's publish another paper, but now we're going to use Y chromosome. Then we publish paternal history of population. And then if you have money, then you say, okay, let's sequence it either full genomes or do SNP chip of those populations for autosomes, which is going to give you even more information, and you publish another paper. But what was my idea is basically to unite all of those markers together into single set of analyses, which I called ancestry packages. So again, we have mitochondria and Y chromosome. We saw that they're very useful for studying human population histories, and depending on which level we are going to cut it, we are going to get different resolutions of differentiations between populations. So again, if you go further in the past, you will see major differences between continental scales, and then if you go further, you might see a differentiation between local scales a bit better. And again, the same applies for the Y chromosome. If we look at the haplogroups that are defined in more ancient times, we will see differentiation only on Africans versus non-Africans. And if we define haplogroups at more recent times, we might see finer scale differences between populations. And in the very same review, they did something that actually inspired me to do this. They started comparing, different types of markers. So here they have autosomal loci. Since this is a study from 2007, they didn't have a lot of data, but nowadays would probably look different. But for the illustration purposes, it's more than enough. They could see that, for example, if we would decide to group individuals on two clusters, we would see differences between two groups. This is wrong, non-East Asian and Oceania. Nowadays, with more data, it would be still African and non-Africans. And then the further they go, they would see finer definition of populations, which might recapitulate how the populations moved across the globe. And they could see actually very similar stories for mitochondria and Y chromosome. And I was asking myself, well, why don't we do that in sort of not only comparative way, just by looking at autosomes versus mitochondria, why don't we put them together in one analysis and get which of the lineages are associated with each other? So to illustrate my idea, let's focus on two populations, blue and brown. And for each of them, we can measure frequency of either autosomal ancestry components here in A, which we would infer, for example, using admixture program, or we would look at mitochondria and Y chromosome, which we can also represent with a color, and those colors are actually representing frequency of certain haplotype. So now imagine that those populations are admixing without sex bias, meaning equal number of males and females are contributing to the admixed population. In this case, 20% of individuals are coming from brown, while 80% from blue. Then if you would measure, estimate frequencies of different genetic markers in this population, we would see basically 80-20 with a little bit of noise around. But when we would start seeing discrepancies between those markers is actually if we would have case of sex bias mating. Imagine our population, blue population, is contributing all of the males and brown, none of them. Then if we would look at a population 4, we would see that all of the Y chromosomes within this population are actually blue, but we would see some different results for autosomes and mitochondria. And if we would now represent these frequencies slightly differently, so let's now focus on the ancestry instead of population. So we would have a blue ancestry here and brown ancestry here per population that are on the y-axis. And the x-axis would measure frequency. So frequency of blue ancestry. And different symbols are going to represent different markers. So mitochondria is going to be in triangle, pluses are Y chromosome, and circles are autosomes. So population 1, we actually constructed it to be only blue. That's why we see only blue. And what we could see for population 4, for example, is that there would be discrepancy between markers. This is just the visualization that I'm going to use in the further slides. So this we can interpret as signal of sex bias processes, where we have discrepancies between Y chromosome and mitochondria. So the main idea could be also to start with given set of populations for which we could run admixture and infer those colors or ancestry components, but we wouldn't be able to classify haplogroups in accordance to those ancestry packages that ancestry components inferred by admixture. We would only have names of the haplogroups, for which usually people in literature, go and describe, oh, this haplogroup that has, 55 letters in it, and it's so hard to follow, is associated with migration from the East or something like that. And it's very time consuming, and often you can get different results. So instead of, going example by example, because someone decided to look only at this haplogroup, let's try to put it all together and see if we are going to see some correlations that we didn't know about before. So the main idea of ancestry packages is to start with data set that would look like this and infer colors for mitochondrial and Y chromosome haplogroups. And in reality, we're often going to have much more many more mitochondrial and Y chromosome haplogroups within each of the populations. And here, for simplicity, I just kept it on 2 or 3 different haplogroups. And once we would classify it, we would be able to investigate sex bias gene flow between different ancestries. So let's focus on the dummy data set, which is basically entirely product of my imagination. So I imagined, what if we would have 3 different continents, brown, blue, and pink continent. And in each of the continents, we would sample 3 populations, where a third population would be somehow product of sex bias gene flow. And that's why we see discrepancies. Would we be able to use such a data set to infer ancestry packages? So this is just for illustration, but in reality, we wouldn't know the colors. So if I would run it, I would be able to first observe the relationship between populations, accounting for all types of markers, and not only autosomes Y and MT, but all together in one. And even more interestingly, I would be able to look at the markers that are driving those differentiations. And I could do that basically using weighted correspondence analysis. And then the output of weighted correspondence analysis I can use in hierarchical clustering on principal components and infer which of those markers that are used to infer relationship between populations are associated with each other. And then I call those as ancestry packages, meaning that, if population is admixing with another population, there should be, set of markers that are co-inherited together. So I defined ancestry packages as those that are having at least one autosomal ancestry component, one mitochondrial haplogroup, and one Y chromosome, but they can have more of them. And let's see now how we would investigate sex bias Again, the same plot, just now we have 3 different ancestries. And if we would see now, population 3, 6, and 9 have actually discrepancies between markers. In this case, we see that, for example, population 9 has more females associated with mitochondria associated with brown cluster and almost none of the males. And this is exactly how we constructed the population. We had only purple Y, which we see here, and mostly mitochondria from brown, which we see here. So it should work. But how it is working for the real data set? So to test it, I actually use it on Southern African populations. Again, the data set is known, but now I included also the autosomal data. I only included additional populations just as a reference to be able to name the ancestry components to either European or whatever. And just for fun, I included also lactase persistence because it was available. Frequencies of this allele were available for the same set of populations. And I wanted to see, we can even use it not only for mitochondrial Y chromosome, but any genetic marker for which we have frequency data. And for that, I had 20 populations. So what I could see is basically, I could infer the relationship between populations, accounting for all markers. And we could see, right, that Bantu populations are closer together, maybe blue ones are all over the plot, red and green are closer together and stuff like that. But what is more interesting, I was able to classify haplogroups into Khoisan and non-Khoisan. And once I did that, I could look at the sex-biased gene flow. And this is a very busy plot, but what you can extract from here is discrepancy between mitochondria and Y chromosome. And we always see more mitochondria that is associated with Khoisans that are in higher frequencies compared to the Y chromosome that is higher for non-Khoisan in almost all of the populations. Again, confirming what I did in my previous study where I showed that there is this pattern. And if I would go a step further and compare it to my diamond plot that I constructed before, now I would have additional depth, which would be frequency of Khoisan or non-Khoisan ancestry on autosomes, and I can plot it now as a color. I could see basically that it's sort of nicely correlating. So grayish ones are going to be in the middle because they're admixed, and the more clear color is, the less admixed they are. And again, we see the same pattern. We have populations leaning towards unions between Khoisan females and Bantu males. And this is how it compares with the original plot. And what is important to mention here is that this is done by dropping the data into R and letting R tell you what is correlated with what, instead of going through literature for months and searching for each haplogroup that I had in data set, what some author 30 years ago said about that haplogroup. Was it associated with Khoisan or not? So I would say this is much more sophisticated way to do than basically going through huge literature search and looking what someone said about haplogroup. But not only that, I could go a step further. So often people are not going to associate haplogroup with very specific ancestry components or population. But now I could go a step further and say, okay, can I see a bit more fine gradient instead of only looking at Khoisan versus non-Khoisan? We know that there are at least 2 admixture events, one involving East Africans and one involving Bantu populations from West Africa, and more recently, probably Eurasian. But this Eurasian might have come also together with East African because of this migration of agriculturalists. And I could basically test if look up through literature and see if my classification makes sense at all. So here I'm just illustrating based on very few haplogroups. For example, Y chromosome haplogroup R1A is indeed in highest frequency in Europe and almost non-occurring in Africa. And I could infer that it's related to West Eurasian autosomal ancestry, even though I didn't include any of European populations in mitochondrial and Y chromosome data set, right? If I would go a step forward, I could see that also lactase persistence, so this is additional marker that I included, called 3910, is in highest frequency in Europe and indeed is associated with the Eurasian cluster. Then very famous example of lactase persistence 14010, which previously has been associated with migration from East Africa, indeed was associated with East Africans. And not only that I could confirm that, okay, haplogroups are belonging where they should, but I also gain some new insight. For example, certain haplogroups that previously couldn't be classified as Khoisan, Bantu, or whatever, now we can classify them. So in my first study, I treated this haplogroup as both Khoisan and non-Khoisan because we didn't know. They are contradicting information in literature, and I could classify it as Bantu actually, but it's involved in heavy sex-biased gene flow. And I'm gonna talk about that just in a second. So I could look at how they're differently distributed across different populations. Again, Khoisan ancestry is mostly predominant in the mitochondria. And what we can see interestingly here is that every other admixture that happened in Khoisans, either 2 or 3 of them, were sex biased, meaning that more males were contributing to the admixture, which is interesting if we think about East African migration, pastoralist migration, that could also have been mediated by males mostly. Same true for the Bantu migration and so on. And if we would focus on a single population, here Ghana, we can see interesting example. Almost most of the mitochondrial lineages that we find in this population are associated with Khoisan cluster, while almost all of the Y chromosome are coming from Bantu. And this is maybe example of strongest sex bias in my study. And interestingly, authors that went to anthropological work, there, they said that this population and others, they believe that they originated as concubinage between Bantu males and Khoisan females. And this, I think, could be a genetic proof that this concubinage actually occurred, which means there is very few males from Bantu population that are intermarrying Khoisan females and making a new tribe, or nowadays even population. And indeed, almost all of the Y chromosomes within this population are belonging to this B2A haplogroup. That was just an interesting thing. So I think that this was showing that uniparental haplogroups are indeed associated with those autosomal ancestry components, and thus we can start thinking about ancestry packages instead of describing separate stories for mitochondria, Y chromosome, and autosomes. That we can use this methodology to actually classify those haplogroups in accordance to autosomes and also geography. We can use it later on to investigate sex-biased gene flow. And this would be probably a necessity to know actually to which population or region certain haplogroups are belonging to. And maybe also interesting is that we can represent unified picture of populationship relationships, accounting for all types of markers, autosomes, mitochondria, and Y chromosome. And directly from the plot, we could read which of those markers are driving differentiations between populations. So those were the conclusions of my thesis. I believe that there is a need to synergetically use all of the markers together, but I would like to do now modeling and see, what are the limitations, how drift is playing the role, or bottlenecks, and so on, to see what are the limitations of this approach to use it for sex bias processes, and also at which level we define haplogroup is the most critical one. Yes. So with this, I would like to thank Mark Stoneking Group. This is Mark Stoneking and all of my colleagues. And of course, you for coming to listen to my talk. And now I would be up for any questions. If you're still there. I'm sorry if it took too long.
So yeah, thanks a lot, Bernard. That was super interesting. Floor is open for questions.
I have some questions. Oh. Thanks for the presentation. Okay, thanks. It was really nice presentation. I liked it a lot.
Thank you.
I have many questions, but maybe just one comment about the lactase persistence. You can also look at skin pigmentation. There is one allele that was also introgressed from Europe like 2,000 years ago into South Africa. So you could also put that in your model.
So I can put anything for which I have data available. So if this SNP is available, for example, on SNP-chip, yes. Or if someone already did it for an entire set of populations that I'm using, that's the limitation.
I see.
But virtually any frequency data that you have for the given set of populations you can use.
I see. Cool. Yeah, so I was interested in how actually the method works. So you have, imagine I have data that I want to use in your package. Do I need extra data to compare it with, or does the package already, because you have data from Africa, right? Like I cannot use it in other populations.
Yes, I think I'm using a wrong term when I say package, because it's not our package yet.
But it's meant to be, or
I would like to make our package out of it, and it's basically very, very primitive and simple. It's running correspondence analysis and HCPC, and that's it. So you can do it yourself. There is no necessity to use it as our package, to make this as our package, because it's very, very simple. But if I would go steps further and include, for example, base trees and possibility to specify at which level you want to call haplogroups, then it would make sense to make a package. And this is my plan for the future, but not now.
I see. Okay, I'll let somebody else ask questions.
Imbar?
That was such a good talk.
Thank you.
I also really like the way that you display things. It's very clear. Do you have a sense of on what time scale this type of analysis could work?
Yes, that's something I'm doing now.
Okay.
So just by doing very basic PopGen drift simulations, it's going to depend heavily on the effective population sizes, right? And on the drift, the strength of the drift. I think there's going to be a lot of noise if you go deeper in the past. If you are trying to infer something that is relatively recent, you will have more power. The further you go into the initial frequency admixture, right? If we like only males from one population and only females from the other, this would, right, give you a bit, it would be easier to detect. So yeah, the timing is going to be dependent on effective population size, ultimately on the drift, right? And on initial frequencies.
And is that sort of noise coming mostly from the autosome part of things because of recombination?
Autosomes I didn't even put in my model. I just use mitochondrial and Y chromosome, and I would say noise should come mostly from mitochondrial and Y chromosome because they have smaller effective population sizes. They do not recombine, and for autosomes you would usually end up. So you could observe, you know, a mitochondrial and Y chromosome as a single SNP, basically, while every other SNP in autosomes is independent, right? So more noise would be associated with mitochondrial by chromosome. And what is even, you know, adding additional level of uncertainty is the number of individuals you're going to sample for males and females, which is a huge issue. You know, if you have 20 individuals and you are calculating haplogroup frequencies, you know, missing one individual by chance, you're going to have like 10% variation or error. So I think this is very, very big problem.
Okay, so you're saying, and would you say that there's basically a sort of a balance between effective population size and time? Or is it basically like back to a certain, the reason I'm asking all of this is because we're going to talk later about trying to do this with lizards. And the divergences are a bit deeper, but I'm like super interested in this. So it's super
yes.
cool.
So what I realized recently is that in many studies, they're using differences between Y chromosome and mtDNA as proof of sex bias gene flow. This is very common in monkeys, for example, or populations that are known to be patrilocal or matrilocal. And I have to say, you know, it can be quite bullshit because you don't know what drift is going to do. So, okay, yes, it's most likely that, but your data has no power to confirm that. So everything that I did could also be bullshit because I didn't do simulations. And we have actually anthropological suggestions that it is probably sex bias. But, you know, there is so much uncertainty with drift that you cannot say, yes, genetics confirms this or not. So I think if times of divergence are beautiful, but if you know that admixture happened recently, so the more divergent they are, the more easier for you would be to distinguish mitochondrial and Y chromosome haplogroups. Thumbs up for that, that's perfect. But if this admixture happened way long in the past, more than 10,000 years ago, and they have small effective population sizes, I would be quite skeptical.
Cool. That's it's amazing and really exciting stuff. And also you've been recorded saying that everybody else's work is bullshit. So good luck.
Oh, thank you. It can be bullshit. I'm not saying it is. We will cut that part. And I also said for my work, just to be sure. Asia, do you want to ask me a question? Sorry.
No, I'm fine.
Carla?
Are you checking if I'm still here or what?
Yeah. Sorry, Carla.
Hello. It was a really nice talk. I learned a lot because I'm not working with some mixture or anything. I was wondering if you will, let's say, if you validate your data to show that you can actually sort of predict like these are mixtures in the past and so on, and you come up with zero package and everything, do you envision also the possibility to then eventually sort of simulate, let's say, what could happen in the future if you will mix certain populations or not really?
No, not what would happen in the future. That's not how it works, right? I mean, you can simulate forward simulations and see like investigate it at different levels, but yeah, I cannot predict because you don't know how populations are going to behave. Are they going to interact with each other or not?
Sure. I mean, it's just assuming certain like.
Yes, but this is what I'm planning.
Modeling exercise, like assuming, okay, if there will be this kind of knockoffs, you need to,
yes.
Modeling.
This is something I have to do now, and reviewers ask for it logically. They need a model, you know, you need to have some kind of guidance when this is true, when it's not true. I mean, I went cherry picking haplogroups, which level I'm going to call it, based on networks. And I see, those haplogroups are occurring in this population, so I'm going to call this as a haplogroup. So it's very problematic. And we need to have, you know, certain threshold or something that can be reproducible to say, okay, I call haplogroup at everything that is defined at 20,000 years ago. And then there is some kind of logic to do it. But yes, simulations are way to go. And also you control, for example, when they are mixing, how much they are mixing. And yeah.
But is there, I mean, models out there you could already sort of recycle or try to use? The reason I'm asking is because I'm not doing that myself, but I know like a lot of people in my institute, they are doing ecological, very advanced ecological models actually, and using also genetic data. For instance, I don't know, with birds. So, I mean, it looks like there is so many models out there that you could try to adapt.
Yes. So the problem is that, again, what people usually do is they look separately for Y chromosomes, separately for mitochondria, separately for autosomes. And when they simulate it, they also simulate it separately. And what I'm interested in is interplay between those, all of those. So when I simulate, I have to simulate all of them at once. And this is not what people usually do. If they're interested in specific questions, such as, I don't know, mitonuclear interactions, they might simulate together autosomes and mitochondria, but not Y chromosome. And now I'm having a student that is trying to do it in SLIM. It's a forward simulator. And I did some preliminary simulations in R that are, yeah, not accounting basically for possibility of evolution, but rather just drift. So there is no, I didn't find a model that I could recycle, basically. I have to do it from the scratch. And I have also to know which parameters I want to use. Effective population size when they're mixing and things like that.
Do you know if anybody's tried this with Neanderthals?
No. No, but I mean, since you're asking the question, I'm assuming you already know the story that is very interesting about
Well, like I
Neanderthals.
know that there's like some admixture of Neanderthal DNA into humans, but I'm wondering if like, I don't know any further than that.
Yes, I thought that you followed the story. So what is weird about Neanderthals is that according to autosomes, Neanderthals are more related to Denisovans. This is even, you know, type of humans that is even further related to us than Neanderthals. But if you look at the Y chromosome and mitochondria, Neanderthals are more closely related to humans, both mitochondria and Y chromosomes. So what happened is probably that there was a mixture of anatomically modern humans leaving Africa and mixing with Neanderthals in some proportion, and subsequently replacing the entire mitochondrial and Y chromosome pool within Neanderthals. And this sounds very crazy, right? But still Neanderthals don't have such a high admixture proportions on autosomes. And this just illustrates how strong drift, or with combination maybe with selection, can be, and how results can be misleading for something that is even, let's say, 200,000 years old and not millions of years old. I think it's very interesting. And yeah, they published also Y chromosome in mitochondria for Neanderthals, but they say it can be explained by slight selection, basically, assuming smaller effective population size for Neanderthals.
Yeah, I guess because I was thinking like probably it would be easier to distinguish the mitochondria or the Y of like different species rather than like populations within a species. And so then maybe even with smaller effective population sizes, you could still get some signal despite the noise and maybe the age. But maybe it's not enough. Or
definitely, the more deeper divergent they are, the easier is going to be for you to pick up the differences for all markers. But it's not true for admixture time between those. Yes. There's something in the chat. You're welcome. You're welcome. I was too long. I talked for 1 hour. I'm sorry for that.
Yeah, I don't know how long this talk is supposed to be, so I just sit and listen.
Thank you for enduring.
It's so cool. So when is this paper coming out?
It's not going to be soon.